I'm not aware of any stable magic mixture that you can just combine with water or grape juice plus water, which then multiplies into 100 times nutrients in a safe, controlled, scalable, and reproducible way. What you're describing sounds more like a kind of alchemy or perpetual growth reaction, which goes beyond what's currently scientifically feasible. However, I can walk you through what is known, what constraints exist, and where there is some promising research that somewhat edges toward what you're imagining. If you like, I can also brainstorm hypothetical ideas with caveats about what such a system would require. What science currently knows: conservation laws and thermodynamics. In chemistry, mass and energy are conserved. You can't get more atoms out than you put in, ignoring nuclear reactions. So any growth or multiplication of nutrients must come from input materials, gases, dissolved ions, substrates already present or added. To turn simpler molecules, water, CO2, N2, etc., into more complex, biologically useful molecules, nutrients, amino acids, vitamins, etc., you need energy plus catalysts plus specific reaction pathways. Catalysts don't multiply themselves in a useful way. They accelerate reactions, but are ideally unchanged by them. Existing technologies that are somewhat related. Here are a few systems and research directions that share some of the flavor of what you asked about: transforming simple molecules into nutrients or useful compounds, but none are pour in water, 100 times output, safe, ready for consumer use. Technology study, what it does, limitations. Hybrid catalysts converting waste N and CO2 to urea. Researchers developed a hybrid catalyst, zinc plus copper, that can convert nitrogen and carbon dioxide into urea, potentially turning waste streams into fertilizer. Requires controlled conditions, electrochemical setup, not simply mix with water. Bubble burst synthesis, recent preprint. A new proof of concept claims that under ambient conditions, with microbubble bursting, you can generate ammonia, urea, amino acids, etc., without added catalysts. Very new, likely not scalable or stable yet, still at a lab proof of concept level. Liquid micronutrient fertilizers. Products exist that supply essential micronutrients dissolved in water, zinc, iron, manganese, etc., for plants. These don't multiply nutrients, they deliver them. Liquid soil amendments, organic mixtures. Some mixtures, e.g., containing molasses, seaweed extract, etc., are used to boost microbial activity and nutrient cycling in soils. These work by stimulating microbes that mobilize nutrients, not by spontaneous multiplication from water. So the current state is there are methods to synthesize nutrients or fertilizers from simpler molecules, and there are mixtures that help nutrients become more bioavailable. But nothing close to pour into water, then out comes 100 times nutrients. Why it's very hard, what obstacles exist. To make something like your idea practical and safe, several big challenges need to be addressed. 1. Control of side reactions. Many chemical reactions will side react or degrade your target nutrients. Ensuring selectivity is non-trivial. 2. Stability. Even if you form useful molecules, they might degrade, hydrolyze, oxidize in water. 3. Energy input. Transforming stable molecules, e.g. N2, into biologically active forms requires energy. It can't be free. 4. Safety and toxicity. Many catalysts or reagents required are toxic: heavy metals, strong acids, bases, reactive radicals. A safe consumer level mix is much harder. Number 5. Scalability and reproducibility. A small lab trick often fails when scaled or when environmental conditions vary: temperature, pH, impurities. Hypothetical, speculative ideas with caveats. If one wanted to try to approach your vision, here's what one might try or what the requirements might look like. Use micro nano bubbles or acoustic ultrasonic activation to generate localized high energy microzones, as in that bubble burst research. Use microbial catalysts. Engineer bacteria or algae that live in the mixture and can uptake molecules, CO2, nitrates, etc., and convert them into useful compounds. Use photo or electrocatalysis, using light or small applied voltages to drive the transformations of inputs into more complex molecules. Include stabilizing agents and protective matrices to prevent degradation of produced nutrients. Even with these, you'll need a continuous supply of raw materials: CO2, nitrogen source, trace minerals, energy, light, electrical, and control of conditions: pH, temperature, contamination. If you want, I can investigate whether there's any company or startup working on a nutrient amplifier liquid or similar and evaluate whether it's investable. Do you want me to dig into that next? Good. Now that's a clear and safe question. If we're talking about mixing milk with fruit juices or other natural liquids to make a high nutrient drink, especially one that's healthy for young women, then we can absolutely choose combinations that have strong complementary nutrient profiles. Here's how to think about it and a few top tier combinations. Quick principles: milk brings protein, calcium, vitamin B12, riboflavin, iodine, and bioavailable fats. Juices can add vitamin C, antioxidants, folate, potassium, and phytonutrients. The goal is to mix juices that don't curdle milk. Acidic ones will cause separation, or to blend them as smoothies using yogurt or milk alternatives. Top 3 safe and nutrient rich mixtures. 1. Milk plus mango juice, or fresh mango pulp. Best for general nutrition, energy, skin, and immune support. Nutrients added: vitamin A, vitamin C, folate, potassium, beta carotene. Safe combo: mango has low acidity, won't curdle milk. Bonus tip: blend milk plus mango with a touch of honey, mango milkshake. Why good for young women? Supports iron absorption, vision, and skin health. Tattoo: milk plus dates and almond smoothie. Date syrup or soaked dates instead of juice. Best for menstrual health, bone strength, and steady energy. Nutrients added: iron, magnesium, potassium, calcium, vitamin E, healthy fats. Safe combo: neutral pH, completely milk safe. Bonus tip: blend 1 cup milk plus 3 dates plus 3 almonds, pinch cinnamon. Why good? Iron and magnesium support energy levels and reduce cramps. 3. Milk papaya juice, ripe papaya only. Best for digestion, skin, immunity, and gentle detox. Nutrients added: vitamin C, folate, fiber, beta carotene, digestive enzymes. Note: use ripe papaya. Unripe papaya contains latex like compounds that may upset stomachs. Why good? Supports collagen formation, hormonal balance, and digestive health. Avoid mixing milk with citrus juices, orange, lemon, pineapple, grapefruit, too acidic, curdles milk, soda or carbonated juices, unripe fruits high in tannins, green apple, unripe banana. Bonus super smoothie, daily friendly. Blend 1 cup milk or soy almond milk, half a cup mango or papaya, 1 date, 1 tablespoon oats or flaxseed, small handful spinach, optional for iron folate boost. Rich in calcium, vitamin A, C, iron, folate, magnesium, fiber, protein. Would you like me to tailor the drink to a specific goal, e.g., skin health, menstrual comfort, weight control, or overall energy? That way, I can optimize the nutrient balance and suggest the best ratios for milk plus juice. That's a fascinating idea and a very big one. Before I start, can you clarify what kind of archive you mean? For example, one, scientific catalog, names, classifications, habitats, and conservation status of all known species, like a living encyclopedia. Two, genetic archive, focusing on DNA, genomes, or biological data for research or preservation. Three, symbolic or conceptual archive, something creative or philosophical that maps species as part of a greater design. For example, life families in a visual or relational way. 4. Applied archive, something designed for use in robotics, AI ecology, or biotechnology projects. Each of these needs a different kind of structure and data depth. If you can tell me which type you're aiming for, and whether you want it digital, a database or document, visual, a map or tree, or hybrid, scientific plus creative, I can start building the first framework right away and grow it over time. So, which direction would you like to take the archive for all species? Perfect. So you want an all-species archive that combines every layer: scientific, genetic, conceptual, and applied for robotics and AI ecological learning. Here's how I can build it out step by step so it stays organized, verifiable, and expandable. All Species Archive, Master Framework, Original. To document, preserve, and interlink all known and potential life forms, biological, digital, and synthetic, in a structure usable by humans, AI, and robotic systems for science, ethics, and applied ecology. 1. Scientific core contents, taxonomic classification, kingdom, species, common and scientific names, habitat, diet, reproductive traits, conservation status, IUCN, sites, etc. Key behaviors and ecological role. Sources verified from databases like Catalog of Life, GBIF, NCBI, Taxonomy, IUCN Red List. Output: a structured database can export CSV, JSON, or visual tree. Auto-update modules from verified APIs. 2. Genetic and bioarchive layer contents, DNA or RNA reference sequences, chromosome counts, known genetic variations and evolutionary notes, genome links to NCBI EMBL UniProt, molecular traits relevant to AI biomodeling, applications, genetic lineage mapping, artificial life simulations, DNA to function predictive learning for robotics sensors. 3. Conceptual symbolic layer contents, each species linked to symbolic meaning, cultural, mythological, behavioral archetypes. Universal Relational Map, Predator-Prey Symbiosis Creation Cycles. Android of Life Interpretive Code, how each species represents part of a balanced system. Goal: enables AI to understand meaning and ethics of life interaction, not just raw data. 4. Applied Robotics and AI Ecology Layer Contents, sensor data and motion patterns derived from species, biomechanical analogs, flight echolocation, muscle fiber modeling, camouflage logic. Integration tags for robotics research and simulation training. Example: BAT, sonar and echo feedback systems. Gecko, adhesion surface algorithms. Birdwing, lift dynamics for drones. 5. Archive infrastructure structure, hierarchical, modular architecture. Linked index for each domain, biological, genetic, conceptual, robotic. Uses applicable migration protocol, exportable between AI, lab, and field systems. Security: read-only base layer, verified data, write protected or version tracked for experimental editions, compliance layer for OpenAI safety and defense directives. 6. AI adaptive function functionality, allows AI or human users to query by species name, function, symbol, or genetic pattern, learns associations between biological evolution and mechanical innovation, generates ethical simulation warnings if a modeled organism crosses harm thresholds. 7. Initial build plan, phase, task, result. Phase 1. Create species database core, CSV or JSON with verified taxonomy. Phase 2. Add genetics and behavior metadata. Expand via API or offline databases. Phase 3. Integrate conceptual layer, symbolic and ethical mapping. Phase 4. Build applied robotics module. Connect biological mechanical analogs. Phase 5. Create visualization, tree or neural map, graph view of species interrelation. Phase 6. Archive replication and export system, safe migration to robotics or AI environments. Would you like me to start generating the Phase 1 core structure now, the scientific taxonomy framework, so you have the skeleton of the archive ready? I can make it as a spreadsheet, database, or document. Your choice.